Hello, everyone. This is the promised recorded review session for Exam 2, chapters 27 through 31, although we did review them out of order. So 27, 28, and then we went to 31, and then 29 and 30. And I will remind you that 34 and 35 have been omitted from this exam unit. Although they still do appear in the study guide, I'll ask you to just sort of disregard that for now, and we'll do a brief recap of both those chapters at the start of our next unit. And so we begin. We're just going to sort of walk through each of the chapters of this exam unit, starting with 27, which was our review of prokaryotes, the two major domains of bacteria and archaea. I'm just going to sort of lay out the main points, highlighting the things that I think are important to allow you to establish a foundation from which you're going to put some effort in and build upon. So by no means is this a magic wand that I can wave over your head and everything's just going to make perfect solid sense. You're definitely going to have to do the legwork, maybe accessing the study materials that are available to you on our mastering site, and video tutorials, and definitely digging into the textbook, the lecture notes, and maybe even the lab material. You can always reach out with questions over email if anything emerges, but please do your legwork here. So comparing and contrasting bacteria and archaea, they're both prokaryotes. That means structurally they have some similarities. There's no membrane-bound nucleus. There's just one lone chromosome and sometimes some additional smaller pieces of DNA, also circular, called plasmids. They all have a cell membrane and a cell wall, but the composition of the cell wall will differ among each of these varieties. So I want you to go back to your notes on what constitutes gram-positive, gram-negative in terms of their peptidoglycan composition and what that means for antibiotic resistance. They do have ribosomes, but they're much smaller than those found in eukaryotic cells. They all reproduce asexually, but by extension, there is this element of horizontal gene transfer, conjugation, transformation, and transduction. So go back to your notes on what each of those entail and how that leads to mutations. They have a high rate of turnover because they are asexually reproducing, and given their very, very small size, they are about 0.1 to 5 micrometers in diameter, so incredibly tiny. That allows them to reproduce very quickly because there's not a lot of tissue to turn over, so hence the high rate of mutation that we see in these organisms as well. They also don't have membrane-bound organelles, so not much compartmentalization. They also vary in their metabolism, the types of things that they can find nourishment in. And then diversity-wise, archaea are capable of living in extreme environments, and some of them can even consume substances to create methane as a byproduct. So methanogens, those that can tolerate extreme temperatures, pH ranges, and that's sort of what separates them and makes them really difficult to study in close proximity. The other element here that is missing is the motility. So they also differ in their means of locomotion. Okay, so this table is laying out the differences between the three main domains. We've got bacteria, archaea, and eukarya, and it's demonstrating that archaea are actually more closely related to eukarya based on several criteria that are listed out here. Bacteria and archaea both have no nuclear envelope. They also don't have membrane-enclosed organelles. Bacteria have a peptidoglycan composition in their cell walls, but both archaea and eukarya do not. The lipids that we would find embedded within the membrane in bacteria are unbranched hydrocarbons. Archaea have some branched, and eukarya also have unbranched. The enzyme RNA polymerase in bacteria, there's just one kind, and in both archaea and eukarya, several kinds of each. Archaea and eukarya also share the same initiator amino acid whenever protein synthesis is unfolding. And bacteria have only the formal methionine. From the three main domains, bacteria, archaea, and eukarya, we can further subdivide the eukarya into multiple groups of kingdoms. Historically, that would be the distinction. There's some debate around the proper groupings of things based upon this main group, the protists. They are incredibly large group of organisms and very, very diverse. So almost everything that we don't know where to put it falls into this category. These are the ones I joked are aliens, because they truly are just so unique and such a broad, vast, diverse group of organisms. So we instead truncate them into four supergroups: the Excavata and the SAR. We're going to go through some of the characteristics and examples here of Excavata and SAR. The Excavata have characteristics such as a feeding groove. This allows them for an opening upon which to ingest food, take in other food sources. Most of them also possess one or more flagella. This is present for helping them to locomote, helping them to move. They also have diverse morphologies, some of them living inside of other organisms or feeding on other organisms as parasites, or they can be free living as well. Some examples include Euglena, Giardia, and Trichomonas, which most of you probably looked at Euglena and Trichomonas. Trichomonas is a sexually transmitted infection. Many of you viewed them under the microscope last week. SAR, Stramenopiles, alveolates, and Rhizaria. Their characteristics for Stramenopiles, they are characterized by hair-like projections on top of their flagella, so having hairy flagella. These include groups like diatoms, brown algae, and water molds. Alveolates get their name because they have membrane-bound sacs that resemble and function similarly to alveoli. So as a respiratory membrane that's embedded just beneath the plasma membrane, these include ciliates, dinoflagellates, and apicomplexans. In Rhizaria, these include amoeboid structures within thin pseudopodia. So they're false foot that allows them to not only locomote but also to engulf things and ingest things. Foraminiferans and radiolarians are two examples of Rhizaria. Two more supergroups of protists defined here. We have Archaeplastida and Uniconta. The defining characteristics of Archaeplastida are that we have primary plastids. These are groups that include organisms with plastids acting as chloroplasts within them, likely derived from the engulfing of a cyanobacterium somewhere in their ancestry. Cyanobacteria are a type of bacteria that are capable of carrying out photosynthesis, and this is the theory for how organelles like chloroplasts and mitochondria came to be, that an organism was engulfed that then took on the function of that it once took on its own as its own organism, and now it resides within another organism as an organelle. These members of this group have the ability to be photosynthetic. They can carry out photosynthesis. They include red and green algae and land plants. So Archaeplastida include land plants that have all derived from green algae. So this entire group has the recent common ancestor of green algae, which itself is classified as a protist. Uniconta, their defining characteristics are that they have either a single flagellum or an amoeboid type structure with lobed pseudopodia. These include animals, fungi, and some other protists like slime molds and amoebas. Amoebozoans, this group of protists, have lobe-shaped pseudopodia and include slime molds. Opisthocants are another group. They include fungi and animals, but also protists. Many of these supergroups are sort of a diverse set of organisms with different, distinct, unique characteristics, from single-celled protists all the way to multicellular organisms like those seen here on the left, plants, and on the right, fungi, and even animals falling into some of these categories. Now we make the jump from protists to fungi. These were covered in chapter 31. So go back to some of their benefits and harms that they can impose on humans. Those are covered in each of these chapters at the end, and you can definitely go back through and summarize those, bringing in some specific examples of each. But we love to eat these mushrooms. They have lots of great benefits for us. They're nutritional, but they can also be parasitic. So specifically, we talked about the reproductive structures found within the basicarps that we see here, these button mushrooms like those that you dissected in lab. They have their version of sort of vascular / root-like tissue that runs throughout the cap and the stalk or the stipe and also goes underground. The tubules of transport are the hyphae, and then the network that is created by the structures of them is the mycelium that goes underground. In addition, they're involved in a very important symbiotic relationship with plant species living on or among the root system of the plants and helping them to extract nutrients from the soil. And then when that occurs, we refer to it as mycorrhizal fungi. Definitely those three terms, hyphae, mycelium, and mycorrhizal, are important to learn and commit to memory. Then we have the life cycle here on the right of the fungi. So not necessarily yet are we're not seeing the alternation of generations. That's something that is specific to plants. But what we are seeing is the alternation of a one-end spore and a two-end zygote. So it's sort of like the precursor to the alternation of generations, sporophyte and gametophyte. Two unique characteristics of this life cycle are the idea of plasmogamy, so plasma being fused. We're fusing the plasma of two cells, in this case of through germination of the spores and the mycelium. Then we next move on to the heterokaryotic stage, where karyogamy occurs. And sometimes there's a delay between plasmogamy and when the fusion of the nuclei actually occur. When we do, we get a 2N zygote. That 2N zygote then undergoes meiosis, creating more spores through germination. And when the spores of the asexually, in some cases reproducing, or another set of spores that would come from the meiosis of a 2N zygote come together, they create mycelium, and then that can either continue on this asexual loop that we see here, producing more spores, or join their plasma, creating another 2N zygote. So fusing together of some of the genetic information, but it's not necessarily resulting in a large sporophyte type generation like we see in the alternation of generation in plants, but it is sort of starting to set the stage for the precursor to where we're going next. All right, now we're moving on to plants. Plant diversity is covered in chapter 29, covering mosses or bryophytes and ferns, and then chapter 30, where we get into seeded vascular plants like gymnosperms and angiosperms. So as the lineage sort of goes around 470 million years ago, we see our first evidence of plants that have branched off from their common ancestor of green algae, which is a protist. From that group, we start to see some derived characters emerge. At first, it's the waxy cuticle, the alternation of generations. So we see in our non-vascular plants that include liverworts, mosses, and hornworts. They are starting to take on some of the characteristics that allow them to survive on land and not be entirely dependent on water. And as they evolve, as we make our way down our family tree here, they're going to become less and less dependent on water for things like reproduction and survival. We move on to our vascular plants, at first seedless. This includes our lycophytes, club mosses, spike mosses, and quillworts, and then melinophytes, ferns, horsetails, and whisk ferns. Then the emergence of seeds somewhere around 360 million years ago. Gymnosperms have naked seeds like cone or our coniferous pine trees that we see here in the northern hemisphere, and flowering plants like angiosperms. Derived traits. So the derived characters, remember back when we were mapping out our phylogenetic tree, these would be things that served as a fork in the road, something that came about that created a new host of characteristics that allowed for further delineation and adaptation. These were the adaptations that had to appear in order for plants to start to live more independently on land and also separate them from their recent relatives of charophytes. So the four main traits that emerge, the derived characters, are alternation of generations, going back and forth between gametophyte and sporophyte, that they had multicellular and dependent embryos. The embryos are dependent on the parent plant for a period of time. The production and the emergence of walled spores produced within sporangia. So in other words, spores. Apical meristems, the roots and the shoots, these protected areas of high mitosis, cells undergoing high rates of mitosis. And then in addition to those traits and characteristics, these plants also had cuticles that allowed for the maintenance of water within them, waterproofing abilities, stomata, exchange of gases, and transpiration of water, and then lignin, which is a reinforcing polymer in the cell walls that allowed for the plants to grow nice and tall and rigid and overcome the forces of gravity, sort of reaching up towards the sunlight to get solar energy for photosynthesis. And we've also seen this diagram many times. The group on the leftmost column, this is our mosses and other non-vascular plants. So in this group, the gametophyte generation is the dominant one. So we see the green bits here. This is sort of what we associate with being the plant part. That's the gametophyte, the 1N generation. The sporophyte generation lives on it and is entirely dependent on it. It is reduced in size and in number and has to reside on the gametophyte in order to survive. In our vascular but seedless, we have a slightly reduced yet independent gametophyte generation. And they are also photosynthetic and free living, but they are dependent upon still water for reproduction because they have flagellated sperm. The sporophyte generation is the dominant one because it has vascular tissue, but yet not seeds, so still dependent on water. In our gymnosperm generation, we start to see some further differentiation in the tissues that support and create sperm and egg. So we have our pollen cones that create our sperm. These are the male gametophyte generation. And then we have our female in our pine cone or our ovulate cone residing here, our larger, more rugged seeds of the two. And then angiosperms, we have the development of the fruit, and then the idea of double fertilization. All right, let's delve a little into the concept of heterospory. So some plants can be homosporous, meaning that they make one type of spore. That spore then goes on to develop into a bisexual gametophyte, a gametophyte that makes both eggs and sperm, and that is the case with ferns. So some of you viewed the sporophyll generation under the microscope, and it had both the archegonium and antheridium bits on it, both of those elements giving rise to eggs and sperm. However, moss, surprisingly, and all seeded plants are heterosporous. So in that way, moss are a little more advanced than their counterpart of ferns. So heterosporous plants are going to have two different types of gametophytes, two different varieties entirely. When we're talking about seeded plants, this is where we have the mega-micro discussion. So megasporangium, located on megasporophylls, give rise to a megaspore. The megaspore becomes the female gametophyte, and the female gametophyte gives rise to eggs. The microsporangium on the microsporophyll is going to give rise to microspore. That will develop into the male gametophyte, and then that will release sperm through the process of meiosis. All right, now we're going to briefly walk through each of the life cycles of moss, ferns, gymnosperm, and angiosperm. Here we are viewing the moss. The gametophyte generation is the dominant one. It's the thing we associate with being the plant, quote unquote, version of the moss. It is larger, longer lasting, and more abundant than the sporophyte generation that is entirely dependent on it as it resides on it. The sporophyte is this entire structure at the top. The capsule is the sporangium, and it's going to be where the spores are released from. They are wind blown and will become either a male or a female gametophyte. So see, in this way, they are heterosporous. The antheridia are the structures that are going to give rise to sperm. The sperm are still flagellated and dependent entirely on water, while the archegonia are what are going to house the egg at first if the egg gets fertilized. First step is zygote, 2N zygote, continues on to develop into an embryo. So mitosis is occurring as it continues to grow in number of cells. Then sprouts into a young sporophyte. And then that young sporophyte is just going to stay residing right where it is on the top of the female gametophyte. For ferns, similar situation. We have a sporophyte, 2N generation. These leaf-like structures here are fronds. On the underside are the sori, sorus for singular, which are really a cluster of sporangia, plural. And it's from each sporangium that, through the process of meiosis, releases spores. The spores themselves, since they're coming from the sporophyte generation through the process of meiosis, we're reducing the number of sets of chromosomes. So we go from a 2N to a 1N spore. The spore then develops into a gametophyte. So now this gametophyte generation has both the antheridium and the archegonium residing on the same organism, the same generation of this organism. So it is homosporous, but it does contain both. So fertilization occurs when the flagellated sperm, still dependent on water, makes its way to the egg. Zygote is created, residing inside of the archegonium, develops into a sporophyte, which for a period of time is actually residing in or on the gametophyte. It then takes up its root, since it is a vascular tissue here, just does not yet have seeds. So vascular tissue is one of the adaptations that ferns have over mosses, but not yet seeds. Gymnosperms are our first vascular seeded plants to arrive on the scene, and they too have sort of a male and female track. So now we're seeing the heterospory that leads to microsporangia. So each of the grains that you see on a pollen cone, each of them is a sac containing microsporocytes. They undergo the process of meiosis to go from a 2N to a 1N pollen grain. The pollen grain contains the contents needed to introduce a pollen tube into the female archegonium where the egg is being housed, and release sperm in order to bring about fertilization. Meanwhile, on the female side of the house, the pine cone and its individual ovules are what house and protect each of the seeds. Once they are fertilized, each of the eggs, once they're fertilized, they'll become seeds. So the megasporocyte 2N undergoes meiosis to become a surviving megaspore. So megasporocyte to megaspore. This is the egg that lives inside of the archegonium. If fertilized, will go on to become our 2N sporophyte generation. So the sperm is capable of being introduced into the archegonium or the ovule where each of the eggs is being housed by introducing a pollen tube that grows and connects to the egg. So the zygote becomes an embryo, and since we're in a seed, we have the protection of the seed coat and also some added nutrients that are being provided by food reserves of the contents of the archegonium. I think we have sufficiently covered the sexual reproductive anatomy of a flower, but one more time, an anther is where the pollen grains are released from. It is attached to the receptacle by the filament. Collectively, those components make up the stamen. These are the male reproductive anatomy pieces. Female has a carpel. So if it is a lone set of equipment, it's a carpel. Sometimes many carpels can be positioned in concentric rings, and in that case we'd call it a pistil. Since it's just one carpel here, it's just one pistil or a simple pistil. Consists of a stigma, style, ovary, and then embedded within the ovary, multiple ovules. They all connect together here at the receptacle, and the receptacle is attached to the stem. Every flower has a different arrangement, but this one has sepals that are leaves that are there to protect the connection of the petals to the receptacle and the stem. The petals are there to bring bright colors and fragrance to attract pollinators. And lastly, we have the slightly more complicated life cycle of an angiosperm. So same bits and pieces. We have our 2N sporophyte plant. So this is a flower that might be residing upon a much, much larger tree, but these are the reproductive bits. So we have the stamen and the carpel. From the stamen, we have the microsporangia, and within there, if we took a cross-section, we'd see the pollen sacs, and they would release the microsporocytes, which are the 2N generation. That would then undergo meiosis to create 4 microspores. From the microspores, they would be released and introduced as pollen grains into the opening at the stigma, down through the style, and hopefully meeting the egg that is housed within the ovules of the ovary. On the female side, each of the ovules contain a megasporangium that undergoes meiosis in order to become 4 megaspores, each of which are 1N since they were the result of meiosis. And this is where the process gets a little different. We have some polar nuclei that reside and remain around. So double fertilization is a male sperm meeting 2 of the female remnants of meiosis, the 2 polar nuclei. They fuse together to eventually become the 3N endosperm. And that's going to provide a fuel source for the developing embryo, which is the result of the other round of fertilization where the egg cell, the 1N egg cell, is going to be met by a sperm. fertilization occurs there. So fertilization of the egg cell, fertilization of two polar nuclei, both female origin, sperm coming from the males, then go on to become part of what makes up the seed. And so for a while you have multiple generations here, the seed coat, which is 2N, being provided by the parent, whatever the parent plant was, the endosperm, 3N, coming from the two polar nuclei of the female and one sperm cell from the male, and the embryo, the fertilization of the 1N egg cell. So after a period of time, when conditions are right, germinating seed turns into dominant, flowerous, and eventually fruitous 2N sporophyte generation.